Hi everyone, and welcome to today's Food Standards Agency Food for Thought seminar on antimicrobial resistance in the food chain. So before we start, just a couple of housekeeping points. We are recording this seminar for upload onto FSA Teams streams for any FSA staff, and for any externals, we're hoping to put this onto the FSA YouTube channel after the seminar. So we very much welcome any questions or comments that you have throughout the seminar. And if you can just drop them into the meeting chat in the comments box as we go through the talk, we'll pick them up at the end and ask our presenters to answer any and field any further questions and discussions. So today we're joined by Ed Haynes, who is a senior scientist at Fera, and Erin Lewis from the FSA's microbiological risk assessment team, who was previously in Ed's research group, which focuses on genomic epidemiology of foodborne pathogens and microbial community analysis, including screening of antimicrobial resistance. So I'd like to hand over to Ed and Erin for our seminar.
Thanks very much, Elspeth, for that nice introduction. Thank you everybody for joining us. Hopefully we'll have a really interesting talk. So yes, as Elspeth said, I'm Ed Haynes. My background is really in the use of high throughput DNA sequencing technologies for questions around food authenticity and food safety. And recently we've been doing quite a lot of work at Fera around antimicrobial resistance, which we'll be talking about today. So if we can move on to the next slide. Thank you. So just for anyone who's not familiar with Fera and doesn't know us, we're a research organisation based just outside York. We were created in 2015 as a joint venture between DEFRA and Capita. Prior to that, we'd been an executive agency of DEFRA. And we've got research groups and scientists working on problems all across the agri-food chain, from crop health to food safety, chemical regulation and environmental monitoring. So really across the whole agri-food chain. We also have a joint institute with Newcastle University that was formed in 2015 as well, called the Institute for Agri-Food Research and Innovation, or IAFRI for short. And this enables us to collaborate really easily with academics at Newcastle, and also we host a number of PhD students at Fera as well, of which Erin was one. So that's all I wanted to say about Fera to start with.
So thank you, Ed. I'm going to take over from here, and I'm going to give you a brief overview of what antimicrobial resistance actually is and what it means to us. So antimicrobial resistance is a natural process where microbes, such as bacteria, but it can also happen in fungi and other microbes, evolve to resist the action of an antimicrobial. So as you can see in this beautiful picture that we have here, antimicrobials can put a selection pressure on population of microbes. So if you can see at the very top in the 0 hours post inoculation, no bacteria are growing on this plate. This plate has an increasing amount of antimicrobials spiked into it. And as we go down, we go on in time, and you can see that the bacteria are stopping at the border and then finding a way to mutate and grow on into the next level, the next increase in antimicrobial content. So basically this allows those bacteria to survive and reproduce, leading to antibiotics becoming less effective over time, and in extreme cases they can become useless. So how do we combat AMR? So there's quite a few ways that we can combat AMR. Obviously, the support of appropriate use and improved control to reduce antimicrobial resistance development, encouraging infection control and the responsible use of antimicrobials are key. We can also invest in the research and development of new diagnostics and treatments, innovation into antibiotics, vaccines and alternative technologies and diagnostics, and obviously collaborating between researchers and industry is a key key part of this, which I think the FSA and Fera both do very well. As well as that, we can improve the access to high quality antibiotics with restricted availability, allowing that everyone gets the right antibiotics at the right doses and duration. So the UK is dedicated to combating AMR, with the AMR National Action Plan being something that the FSA is involved in quite heavily, as are a lot of departments in the UK government. But how does AMR actually happen? So there are two key mechanisms for antimicrobial resistance. The first is, as you can see in this diagram, things like efflux pumps or enzymes. So essentially what you want is to stop the antimicrobial from reaching its target in a high enough concentration that it can actually be effective. So a good example of this is with efflux pumps, where they may already be present in the bacteria, but if the bacteria decides to upregulate, so increase the production of these pumps, it can actually pump the antimicrobials back out of the bacterial cell, which leads to a lower concentration of the antibiotic inside the cell. That means that essentially you've not got enough antimicrobial in the cell to kill it, and so you can get resistance that way. The second way is to modify or bypass the target of that antimicrobial. So that essentially means making something slightly differently to the way it would have made it before. So things like proteins in the cell. A good example of this is by expressing alternate proteins. Some microbes are able to produce these alternative proteins. For example, in MRSA, which is a resistant form of Staphylococcus aureus, which you are probably aware of. It's often known as the superbug and found in hospitals. This resistance is caused by a gene called MEK-A. It's an alternative protein which essentially has a lower affinity for beta-lactam antibiotics, and therefore it confers resistance. So those mechanisms are kind of a brief overview of what can happen, but how does that actually happen in real life? So antibiotic resistance primarily occurs through either the mutation of genes that are already in the bacteria or fungi, or through acquisition of new genetic material that leads to the change in the antibiotic response. The selection pressures caused by the use of antimicrobials can speed up this process and the process of an entire population acquiring resistance. So as you can see in this CDC picture here, if you have lots of germs and a few of them are resistant to antibiotics, when we give the antibiotics to kill the bacteria, it also kills everything else in our bodies. And the antibiotic resistant bacteria can grow and take over. In addition to that, the selection pressure caused by antimicrobial resistance, caused by antimicrobial compounds, can lead to some bacteria giving those genes to other bacteria. So more of them can survive. And it can also transfer those genes to new bacteria that wouldn't have had those before. So how do we test for antimicrobial resistance? So using traditional microbiological techniques, as you can see here, it will be our disc diffusion method. So basically we put the bacteria onto a plate, we put circles of paper infused with our antimicrobial compound, and we see how well the bacteria can grow in the presence of the antimicrobial. So if you look at part A of this picture, A and D are preventing the bacteria from growing up towards them, as are G and H. B seems to be causing some decrease in the amount of bacteria that's growing up to it, but not all the bacteria is resistant, and C, E, and F doesn't seem to cause any decrease in the amount of bacteria on the plate. So we would say that in this case, probably A and D would be susceptible antibiotics, or bacteria are susceptible to these antibiotics, as are G and H. But for C, B, E, and F, it's likely that they're resistant. Another way that we can measure antimicrobial resistance is by looking at the minimum inhibitory concentration. So this is a similar method, but instead of using discs containing a known concentration, we use a strip that contains an increasing or decreasing concentration of the same antimicrobial compound. So as in picture B here, you can see that this bacteria is unable to grow up until where that nice red line is. So that would be our minimum inhibitory concentration. So basically the amount of drug or antimicrobial compound that you would need to give to get rid of this bacteria. So this allows for the determination of phenotype. What that means is that essentially we know that these specific bacteria are specifically susceptible or resistant to certain antimicrobial compounds. It also allows for attribution to a specific organism. So we know that what we put on that plate is whatever we put on that plate. So we know that that specific organism is resistant to specific antimicrobial compounds. Another way we can screen for antimicrobial resistance is using next generation sequencing. Now, there are two potential methods for screening of antimicrobial resistance using NGS. The first is whole genome sequencing. This is where we isolate a pure culture of an organism of interest, for example, listeria, and sequence all of its DNA. This will allow us to find antimicrobial resistance genes in that specific bacteria. The second method is through metagenomics or metatranscriptomics. This is where we sequence the total DNA or RNA in a sample of mixed bacteria and fungi. So if we look into further detail on metagenomics versus metatranscriptomics, metagenomics sequences all the DNA in the samples. It detects the presence of AMR genes or mutations and is increasingly being used in a public health setting. Metatranscriptomics sequences all the RNA in our sample. Both these methods have problems with host contamination, but metatranscriptomics has some methods available to enrich the microbiome. So that's the part of the RNA that's associated with the bacteria and fungi and germs in your sample. And it's also the only method that can detect RNA viruses, which for food microbiologists is quite important because we have things like norovirus to consider. It detects the presence of RNA transcribed from AMR genes or mutated genes. And this is a potential advantage because it means that the gene is not only present in the genome of this bacteria, but it also potentially is active at the moment of the extraction. So it's being transcribed, so it's having proteins made, so it's likely that it might infer a phenotype. So what are the pros and cons of using AMR, using NGS to detect AMR? Well, when we use whole genome sequencing, we can screen for multiple and possibly all known genes. And we also get a whole raft more information that allows us to look at things like source tracking and also other genetic information on that bacteria. The key downside to whole genome sequencing is it can't give us information on the phenotype or the expression of genes. So we don't actually know, even if the gene's present, whether it would infer a phenotype. It's also quite expensive compared to traditional methods. The pros and cons of metagenomics or metatranscriptomics is, again, we can screen for multiple, possibly all known AMR genes, it also removes the need for isolation, plating, or cloning that you would need for both whole genome sequencing and traditional characterization methods, and therefore there's less time input needed. It also gives us information on the microbiome of the sample, so that's all the other organisms that are present in the sample, which can give us a lot of good information. The key downsides to this method, though, is it, again, is not easy to get information on the phenotype or viability of the organisms. It's also difficult to assign specific AMR genes to a specific organism in the sample. So just because we have the gene for antibiotic resistance and listeria in the sample doesn't mean that that specific antimicrobial resistance gene comes from the listeria. It could come from anything in that sample. And in addition, we can't be certain that the organisms are found aren't false positives. The way that we assign organisms to the DNA sequences is prone to error at times, and this is a fairly new technique, and so some of the applications are slightly harder to use. So as part of my PhD, I looked at the applications of next generation sequencing for the assessment of microbiological safety of fresh produce. My first chapter was a method development chapter, looking at whether we can use NGS and bioinformatics in the analysis of food samples. My second chapter looked at NGS of the fresh produce microbiome, and the third, phenotypic and genotypic screening of Listeria monocytogenes, isolated from fresh produce. So why fresh produce? Well, the World Health Organization estimated that there were 600 million cases of foodborne illness worldwide, and estimates of about 10% of those being attributed to fresh produce. And there are large, high-profile outbreaks that are still occurring that are associated with fresh produce. In addition, most illnesses attributed to fresh fruit and veg are believed to be caused by contamination of the food with feces, and prevention methods. Prevention of disease is most often due to prevention of contamination, because decontamination and detection techniques are still fairly difficult and often can't be done without tainting the product or within the shelf life of the product. So the first chapter of my PhD looked at methods of NGS to look at the microbiome of fresh produce. I did find in this chapter that the limit of detection for NGS is dependent on both sequencing method and analysis technique used, and that all samples that I sequenced were a high proportion chloroplast because of the host contamination that I talked about previously. As well as that, and the key finding that I wanted to highlight here is that all limits of detection for all the methods that I tested were less sensitive than the current ISO methods, and less sensitive than the concentrations of pathogens usually reported for fresh produce. So to try and combat this, I focused on increasing the sequencing depth in my second chapter, where I essentially did Metatranscriptomics of the fresh produce from the food supply chain. So the key findings that I wanted to highlight today were that of the fresh produce, the 50 fresh produce samples that I screened, I only actually found 4 AMR associated genes, and none of these were genes that were to medically important antimicrobials. As well as that, we found that when compared to traditional microbiological screens, there were some discrepancies between Enterobacteriaceae and Listeria species that were found using NGS versus traditional microbiological techniques. And in addition, the top genre which we found associated with the produce varied by produce type, although Alteramonas, Pseudomonas, Leuconostoc and Rhinella, all of which are spoilage organisms, were the top ones found in all produce types. So produce type does have a strong effect on the microbiome, and Additionally, there's only essentially there's very low levels of AMR found in fresh produce in UK fresh produce from the supply chain. So I'm going to hand back over to Ed now.
Thanks so much for that, Erin. I'm going to talk now briefly about two projects that we've been working on for the FSA that use really similar metagenomic techniques to those that Erin has discussed. And the first project is one around the burden of AMR genes in ready-to-eat foods. So if we look at the next slide. So as we've discussed, antimicrobial resistance is a potentially big problem. The use of antimicrobials in the food chain can lead to the evolution of AMR, and this has the potential to be transferred into human consumers when they eat foodstuffs. Ready-to-eat foods as a group are also, if we go back to, yeah, there we go. Ready-to-eat foods as a group are of particular interest as well because they're consumed without any further cooking in the home, meaning that any AMR bacteria or AMR genes that are present on these ready-to-eat foods might be ingested and taken into the host consumer microbiome. And they also span a range of different production techniques, from dairy produce to fresh produce, vegetables, that have very different inputs. So within this broad category of ready-to-eat foods, there might be really different drivers of AMR evolution. So next slide, brilliant. So we were trying to use metagenomics to understand the AMR burden of ready-to-eat foods in the UK. So as our sampling strategy, we selected 1,000 samples to cover around 90% of total UK consumption across non-complex ready-to-eat foods. And that figure at the bottom just shows the number of different samples for each different food type. When we say we're interested in non-complex food, what I mean is that for ready-to-eat foods such as pre-made salads or pre-made sandwiches, we didn't want to investigate these because if we found AMR genes in these food types, we wouldn't know necessarily which ingredient those AMR genes were associated with within that complex mixture. So it would not be massively informative in terms of where the AMR risk lay. But neither did we want to restrict ourselves to purely single ingredient foods, because that would have excluded a lot of really highly consumed ready-to-eat foods. So we've got things like cheese and ham in there, which are primarily a single ingredient, but they may have some small levels of additives like salts or preservatives. So this all gave us a total of 52 different sample types to look at in total. And as you can see from the graph, they're dominated really by milk of various kinds. Although there are some fresh produce types that are quite highly consumed, bananas and apples, for example. There were relatively few ready-to-eat meats that were consumed in high enough levels to form part of our study, really only ham and corned beef, I think. We'd also originally planned to look at ready-to-eat seafood as well. But when we looked at the consumption data, ready-to-eat seafood was so low in consumption in comparison to dairy, produce and meats that we decided to focus primarily on the most consumed foodstuffs and sample according to consumption. So once we had our sampling strategy, samples were then taken from retail sale around the UK according to population size, and then from supermarkets based on their market share by Hallmark Veterinary and Compliance Services, who were working with us on this project. So the next slide. So each of these 1,000 samples were couriered back to Fera, and when they were received by us, where possible, samples were rinsed. And then, so the outside of them was rinsed with a rinsing buffer, and DNA extraction was then performed on that rinsate. And this is really to do with exactly what Erin was discussing earlier about trying to reduce the amount of host contamination in our samples. So we've got a limited amount of sequence or sequencing power available for each sample, and we don't want to use that up sequencing the ingredients. We want to try and focus on the microbiome. But there are some food types where it's just not possible. Milk, for example, is very difficult to rinse. So we just did total DNA extractions on those product types. So when we extracted those 1,000 samples, we then sequenced them using an NGS technique that is suitable for profiling bacterial communities, which is 16S metabarcoding, which we can talk about later if it's of interest. And the reason that we did that was to determine two things, really. One was whether there were detectable levels of bacterial DNA present. So did the sample yield enough bacterial DNA to be worth taking forwards? And two, to look at whether the sample was dominated by host DNA. And we were able to do this by exploiting a bit of a quirk in the way that 16S metabarcoding can work, is that as well as focusing on bacterial sequence, it also can provide sequence from host mitochondria or chloroplasts. And this gives a bit of a qualitative indication about the degree of host contamination of our samples. So when we've done all this, we then selected 256 of our samples, again according to consumption, but weighted towards sample types that had a higher proportion of bacterial DNA, and took these forward for full metagenomic sequencing. So on the next slide, we can see this is just an example of the sorts of outputs we were getting from the community sequencing approach. The pie chart shows the top 5 bacterial taxa or bacterial groups that were identified in each of these sample types. On the left pie, we have apples, which we are able to rinse. And when we extract the rinsate from the surface of those apples, we get primarily dominated by different bacterial groups and a little amount of chloroplast sequence there. On the right pie chart, we have apple juice, which we have to do a total DNA extraction from. And there you can see the sample is really dominated by chloroplast sequence. So as you would expect. So on the next slide, you can see some preliminary analysis. So we've done several rounds of analysis on the high throughput metagenomic sequencing data, but exactly as Erin said earlier, we're seeing some issues with potential false positive sequences. So maybe short or non-complex sequences in the AMR gene database are matching with some of our sequences. So in the next couple of weeks, we hope to have decided some ways forward to remove those, but in the interim, I can show some preliminary data. This figure here is, oh, sorry, Erin, if we go back. This figure shows, kind of shows the effect of the rinsing technique that I was talking about earlier. So on the x-axis, we've got number of reads post QC. That's really the number of high quality DNA sequences that came out of the machine for each sample. And on the y-axis, the vertical axis, we've got the number of sequences that are left after all of the host sequences that are taken out. So the number of microbial DNA sequences. And it's a bit of a varied picture, but you can generally see that for the blue circles, for the produce types, where we generally are able to rinse, we get a lot more bacterial sequences coming off. And for the dairy types, for example, in red, where we've got, we're usually having to take total DNA extractions, we get a lot more host sequence or a lot less microbial sequence coming out, which is exactly what we would expect, really. So on the next slide, we've got just, this is just an example of the sorts of data that we've generated so far, although, as I said, this is all subject to the caveats around just the final kind of QC of the data. On the bottom, you've got all of the different food categories we've got. And on the y-axis, we've got a normalized number for the number of different sequences from each sample that map to or that are similar to any AMR gene. So just a kind of average measure of AMR, amount of AMR gene sequence in each sample. And you can see these box plots show you kind of the average number of AMR genes per sample and the spread within a sample type. And you can see that some sample types, maybe around the dairy, appear to have quite high average numbers of AMR genes per sample and quite high spread. And some other produce types have lower average levels and a lower spread. But as I said, this is all preliminary, but we should have some final numbers in the near future. So the other project that I want to mention really briefly at this point is just around, it's a newish project that we've started for FSA around AMR in biofilms. formed during secondary meat processing of meat products. So on the next slide, if we look at that, we see that bacterial biofilms are essentially formed when bacteria secrete long polymer molecules out of their cells into the environment, and these long sticky molecules stick bacterial cells together and also stick them to environmental surfaces and provide them with a degree of protection from chemical or mechanical cleaning. So these biofilms may allow bacteria to persist in food processing environments. They might also increase the risk of AMR by allowing bacteria to survive exposure to different biocides in the processing environment and then evolve resistance. And furthermore, bacteria in these biofilms are held in quite close physical proximity, and that might enable the exchange of AMR genes between different bacteria. But there is a bit of an evidence gap as to how much biofilms do contribute to AMR contamination in foodstuffs. So in this project, we'll be taking biofilm samples from around different secondary meat processing plants, bring them back to Fera and sequencing them in the same sorts of techniques that we did in the previous project to try and identify any AMR genes that are present and work out how important or otherwise biofilms are. So back to you now, Erin.
Yeah, thank you, Ed. So yes, as Ed said, some great uses of metagenomics in the detection of antimicrobial resistance there. I'm now moving forward to the third chapter of my PhD, which was looking at phenotypic and genotypic screens of Listeria monocytogenes isolated from fresh produce. So Listeria, I mean, we're all, well, most of us here are going to be food microbiologists, so I would, well, some of us here are food microbiologists, so I would hope that we know of Listeria. So Listeria monocytogenes is a key issue because it grows well in numerous environments, but also at low temperatures. So Listeria can grow in refrigeration temperatures, and so that makes it a key issue for food and foodborne disease. Most cases of Listeria are due to contaminated food, so it's actually quite a high impact area for the FSA, where we could potentially make quite a lot of difference. As well as that, a high percentage of the hospitalisations in the UK that come from foodborne diseases are due to listeriosis, a severe form of Listeria infection, with an up to 20% mortality rate for these cases. And of course, the key treatment for Listeria monocytogenes infection is antibiotics, hence the need to be aware of the antimicrobial resistance burden in this organism. So, antibiotic resistance, as part of my PhD, I wanted to look at the antimicrobial resistance profile of Listeria. Obviously, we've been through what antibiotic resistance is, and that they can pass between bacteria and they can also be acquired from the environment and potentially confer resistance to multiple antimicrobials. As well as that, in this chapter, I looked at the potential virulence of Listeria monocytogenes. So virulence are essentially how easily Listeria or an organism can infect its host and how severe the disease will be in the host once it is infected. And we can look at these in a similar way we can to antimicrobial resistance, because there are some genes that are known to us that will increase the virulence. As well as that, as Ed has given us that nice introduction into biofilms, I also looked at the ability of Listeria to form biofilms. As Ed said, this can decrease the efficacy of both biocides and antibiotics and lead to enhanced survival of these organisms. So this chapter essentially was split into two parts. The first part was a genotypic screen. So I did whole genome sequencing, as I explained earlier. I took the isolates from vegetable origin. So I had a collaborator who was screening vegetables for us and sending me isolates of Listeria monocytogenes from vegetables in the food supply chain. And I was running whole genome sequencing on these before analysing them. As well as this, I also did some traditional phenotypic screening in the lab. The first screen that I did was a biofilm screen. So essentially, I had 24 well plates that I inoculated with Listeria. I let them grow for 48 hours, and then I washed them and stained what was left behind. What was left behind was essentially the biofilm of Listeria that was connected or attached to the bottom of the plate. And that allowed me to gain a quantitative measure of exactly how much biofilm was created by Listeria, different Listeria isolates from within the food chain. The second screen that I did was an antibiotics resistance screen. So as I showed earlier, this was using the disk diffusion method. I had 5 different antibiotics that I screened for. These were in line with the EU guidelines on what we would traditionally screen for in Listeria. So it was benzylpenicillin, ampicillin, meropenem, erythromycin, and I'm not even going to try and pronounce that final one. In fact, trimethoprim sulfur methoxyl. I'm sure I got that wrong, but yeah. So we screened for those 5 antibiotics on the 50 isolates from vegetable origin. So using the phenotypic testing, so that was the isolate in the lab, we found that for the 5 different antibiotics that I've mentioned before, none of the isolates that I tested were resistant. All of them, there was variability found between isolates, but all of them were above the EU guidelines for what would be deemed resistant versus non-resistant. The biofilm screens were much more interesting, and to be honest, every single Listeria isolate that I tested was able to form a biofilm, but it did vary quite considerably between Listeria, different strains of Listeria. And additionally, I did the biofilm screens in both NB, which is nutrient broth, which is quite a tough substrate for Listeria to grow in. It doesn't really like being there. And also grew them in brain-heart infusion, which is a very rich media. Listeria loves it. It's what it would like to grow in. And I found that in the nutrient broth, Listeria, when it was challenged, was able to grow biofilms better than in the brain-heart infusion where it wasn't challenged, suggesting that in the food chain where they are being challenged, they might be better at forming biofilms. And using the genotypic methods, I screened again the total resistome and found that all isolates were actually resistant to FOS-X and Lin, which is unsurprising as it has been noted in the literature that basically this is a ubiquitous gene that is found in all Listeria. 2 vegetable, 1 meat and 1 clinical isolate. The meat and clinical isolates I obtained from a public database also had resistance to BCRB and BCRC genes. This was interesting as they're not traditionally screened for because they're not used for treatment of human illness, but they are often used, compounds that are often used in things like antimicrobial hand washes or antimicrobial hand gels. I also looked at the potential virulence of these isolates. I found 42 genes, but they were not specific to a single category of origin. So whether it was from vegetables, meat, or clinical isolates, you couldn't tell the difference. So I'm going to hand back over to Ed now.
Thanks very much, Erin. The last project that I'm going to talk about now is actually a project that was jointly funded by FSA and by DEFRA. And this was a review of antibiotic use in crops and the associated risks of AMR. So if we go to the next slide. So the use of antibiotics in agriculture can drive the evolution of AMR, but the vast majority of work done to date on AMR in agriculture has been around livestock agriculture, which makes perfect sense. Livestock are treated with antibiotics to treat infectious diseases, and also in some countries at some times antibiotics have been used for other purposes, such as growth promoters. But there were a number of reports in the popular press recently about potential antibiotic use and misuse in crop agriculture. For example, reports of trunk injections of amoxicillin in citrus in Thailand, approval of streptomycin and oxytetracycline for citrus in the US. And this prompted an investigation of the risk of AMR in crops. So we undertook a desk-based study to try and establish some baseline knowledge about the extent of antibiotic use and AMR in crops globally. And the sorts of questions that we were trying to address were what crop diseases are being controlled by antibiotics and where, what do we know about antibiotic presence in the crop environment, potentially from other sources than direct application, what technologies are available for detecting antibiotics and AMR, whether any other agrochemicals might be responsible for AMR evolution as well, and what knowledge gaps there are. And this was all restricted to primary production, so agriculture. Secondary processing of food products was out of scope of the review. And we also focused, as we have in all these projects, on antibiotic resistance in bacteria rather than in other groups. So the way we gathered information for this desk study was primarily through a quite traditional kind of literature survey of published and grey literature. But we also tried to supplement this with expert opinion solicitation to try and obtain information about antibiotic use that wasn't present in any literature, but experts might know about. So to that end, we sent a questionnaire out to any plant health contacts we had and International Plant Protection Convention national contact points in a variety of countries, selected based on whether we had relevant contacts and assessment of how likely they were to be important as exporters to the UK of crop-based foods, and to try and ensure a broad geographical spread. But we did get a very limited response, I would say. So this map here shows in orange, you can see the countries that we asked and sent the questionnaire out to and didn't get a response from. And in blue, you can see the countries that we did get a response from. So the 6 countries that did respond to us were Germany, Kazakhstan, Kyrgyzstan, Rwanda, New Zealand and Zambia. So thank you very much to them for responding. We also asked EPO, the European and Medical Plant Protection Organisation, and EFSA for information, and we got in contact with CRD in the UK, and they did provide us with some useful information as well. So that information was synthesised with information available in the published and grey literature, and it's a bit of a monster table, and there are some much bigger and more in-depth tables in the final report that should be made public and available, I think, very soon, in the very near future. But this table just kind of summarises what we know about what crops antibiotics are being used on and what diseases they're being used against. I should point out, one, streptocycline, SC, isn't an antibiotic, but a 9-to-1 mixture of streptomycin and tetracycline. But the grey literature reports did suggest this was quite widely used in some South Asian countries in high doses and on a range of crops. And also a recent paper by Chanvatik et al. Has produced evidence that more antibiotics, ampicillin, amoxicillin, streptomycin and penicillin are being used in Thailand in mandarin production. So more evidence is emerging all the time. So on the next slide, we've got just a kind of summary overview of what we found about the direct use of antibiotics. So primarily the use of antibiotics in plant agriculture is to control bacterial diseases of pome fruits like apple, stone fruits, citrus fruits and sometimes vegetables. And the largest recorded uses are on apples to treat fire blight, and potentially now on citrus to treat citrus greening in the US. But resistance has definitely arisen, so there is documented evidence of streptomycin resistance in apple orchards that have been treated with streptomycin. But the timing of antibiotic treatment can reduce or eliminate the presence of antibiotics in the actual products, in the fruits themselves. So in the streptomycin example, streptomycin is applied during flowering. So by the time the fruit has ripened and is ready for harvest, the antibiotic itself will have degraded, but any antibiotic genes, antibiotic resistance genes or any antibiotic resistant bacteria that have evolved may still be present. There has been some really interesting work after we published the review around, or after we finished the review, around recommendations from plant health outreach workers, so-called plant doctors, in low and middle income countries for different plant protection products for different diseases. And this has shown that rice is actually a crop that has the highest proportion of recommendations for antibiotic use. So we did find evidence in our review as well of rice being treated with antibiotics. And this other review also found that antibiotics, they found widespread use of, or widespread recommendation of streptomycin and tetracycline combinations, just like we did. Other examples of inappropriate use against fungi or invertebrates, and no evidence of reduction in antibiotic recommendation over time, which is really interesting given what we think of as this global awareness of trying to reduce antibiotic usage. So really, to kind of summarise, where there is some direct evidence of antibiotic use in crop agriculture in countries with strong regulation and good documentation, it appears quite low, certainly lower than in livestock agriculture. However, where regulations are weak or non-existent, use may be really quite widespread. And there are other alternative sources of antibiotics in crops, as well as direct application for treating plant diseases. So manure, sewage, grey water are potentially important sources of antibiotics in crop agriculture. And in countries with strong regulations around the use of antibiotics in crops, these are probably more important sources than actual direct application. Antibiotics and AMR genes in the crop environment are readily taken up into plants. But this is highly variable based on lots of different factors like compounds, plant type or soil type. We did find that evidence that the uptake of antibiotics into plants is much greater in hydroponic systems than in soil-based systems. Beyond antibiotics themselves as drivers, one thing that I thought was really interesting that we looked at was potential co-selection of antibiotic resistance. So I've lumped a few different phenomena under the term co-selection, but this is essentially where chemicals other than antibiotics induce resistance in bacteria to antibiotics. And there are a number of different mechanisms whereby that might occur. So one could be cross-resistance. So this is where the same mechanism supplies resistance to more than one different compound. So this could be like an efflux pump that Erin was mentioning earlier, that pumps out both a biocide or an antibiotic. So a biocide could induce evolution of resistance to antibiotic. Or there's co-selection, where genes that confer resistance to antibiotics and other agents are located in the same mobile genetic elements. These could be different genes on the same plasmid that are then co-inherited or co-passed around different bacteria and co-selected for. Or there's also a phenomenon of adaptive resistance, which is where resistance to antibiotics can increase due to differences in gene expression caused by environmental variation. So this could be where the presence of a pesticide could temporarily make a bacteria more resistant to an antibiotic. And this could then lead to long-term resistance to that antibiotic if the bacteria can tolerate high levels of that antibiotic and evolve resistance itself. So for adaptive resistance to take place, you would need both the presence of the driver and the presence of the antibiotic at the same time. So to really kind of briefly summarize what we found, there is extensive evidence that metals can co-select for AMR. This has been known about for quite a while in a number of different kind of areas, such as industrial pollution. It's quite a well-known example. From an agricultural point of view, copper is almost certainly the most important metal that could be co-selecting, and that there's evidence that co-selection occurs. So this could enter the agricultural environment for a number of ways. So copper is used as a fungicide in crops like vineyards, olive groves and orchards quite widely in Europe. It could also enter the crop environment through pig manure. So copper can be added to the pig diet as a growth enhancer, and a lot of that can then be excreted out in the feces and then applied to the land. Or atmospheric deposition is another source of copper in agricultural environments. So this could be quite important. But as well as metals, we also found pretty strong evidence that some pesticides, such as glyphosate, can lead to increased rates of AMR, and this is almost entirely through adaptive resistance. So the pesticides temporarily making bacteria more resistant to different antibiotics. Although there was some evidence that other mechanisms could be taking place, there was evidence that an enzyme conferring resistance to a particular insecticide could also potentially be directly providing resistance to antibiotics by degrading them. And the take home here is really that very few classes of pesticides have actually been assessed for their ability to co-select for AMR at all. And finally, there was some evidence found that organic pollutants like polyaromatic hydrocarbons or polychlorinated biphenyls and some biocides could potentially co-select for resistance as well, or at least shift the soil microbiome to a community that was more resistant. But from a crop agriculture point of view, these are possibly less important than metals or pesticides. In terms of what we found around detection methods, I should say off the bat, I am not a chemist by any stretch of the imagination, so I will be unlikely to be able to explain anything about mass spectrometry. But my colleagues did find that there are a number of published methods available to detect antibiotics in different crop products, different types of fresh produce, but these are quite limited compared with those that are available for animal tissue. And they also found that the role of non-targeted approaches using high-resolution mass spectrometers to detect many thousand different chemical compounds at the same time are going to become more and more important. In terms of AMR detection, and this could be a whole review and seminar in itself. There are many different targeted assays for different AMR resistance genes and different ways of measuring AMR itself, and it was kind of beyond the scope of the review to list them all. But what we did do was try and provide examples of novel approaches that are being developed to try and address the limitations of previous approaches. So, for example, around phenotyping, looking at actual bacterial physical resistance to an antibiotic. As Erin said, the traditional way of doing that has been by plating out bacteria on agar plates, but researchers are developing methods to try and phenotype individual bacterial cells, for example, by hydrodynamically trapping cells and then microscopically examining their motility and morphology to different antibiotics. And this has the benefit of allowing you to look at potentially mixed populations of resistant and susceptible cells, and also it's actually quicker to monitor the response of individual bacteria than to wait for them to grow into visible lawns on agar plates. Targeted molecular assays to look at antibiotic resistance genes. As I said, there are many of them, primarily based on different PCR approaches. LAMP stands for loop mediated isothermal amplification, and it's a method that is it's an amplification method for nucleic acid targets that is kind of similar to PCR, and it amplifies up a known target, but it's more suitable for use in the field than PCR, which may be relevant from a crop point of view. And as well as detecting genes, people have also used it to try and infer phenotypic resistance, for example, by testing cultures that have and haven't been subjected to antibiotics, or by trying to detect messenger RNA, mRNA, to look at genes that are being actively transcribed on exposure to antibiotics. And then the non-targeted approaches are really very firmly based on the sorts of metagenomic and metatranscriptomic approaches that we've discussed at length in this talk. So really just to summarise where we thought there were kind of knowledge gaps and further work that could be done, There are a number of technical improvements that could be made to antibiotic detection methods. A greater understanding of antibiotic metabolism or bioaccumulation in plants might help us understand practices or products that might be particularly risky or otherwise. Like I said earlier, a lot of agrochemicals haven't been assessed for their ability to co-select AMR. Given our limited response to our questionnaire, some kind of information sharing exercise or network to try and encourage sharing about information about antibiotic use in crops could be useful. And really, given the relatively low baseline knowledge about antibiotic use in some countries, you could take some kind of surveillance approach to look for antibiotics or their breakdown products or AMR genes in crops that are imported from either large-scale supply countries or countries where misuse might be suspected, or both. So that's really a summary of what we found in that review. I'll go back to you, Erin, now.
Yeah, thank you very much, Ed. So Ed's obviously given us a great overview of some of the FSA AMR projects that are ongoing at the moment. I wouldn't be an FSA employee if I didn't plug a couple more while I'm here. So these surveys, the ongoing FSA research can kind of be lumped into a few different areas. So at the moment we've got ongoing literature reviews. So as Ed's spoken about, we've got a few of those ongoing. We've also got a couple of EU surveys and a survey of Campylobacter from retail chicken in the UK. In addition to that, we have a fairly large portfolio of research projects, some of which are coming to an end as we speak, others are just starting. And in addition, we have modelling projects, one modelling the risk of antimicrobial resistance within the food chain, and